in the acceptor stem region, which is important for the formulation of the tRNA. But of course, formulation is not there in archaea and eukaryotes. The other region is the 3 GC base pairs, consecutive GC base pairs, which are really highly conserved in all domains of life. And so this turned out to be something which one would want to follow a little bit more to understand how proteins or how initiation happens. Now, one of the things that I did in Tom's lab right in my beginning when I joined was to set up this in vivo initiation as a system. And the SS system in retrospect is very straightforward. We have the reporter gene, chloramphenicol transferase. All I had to do was change the start codon from AUG to UAG and make a corresponding change in the anticodon of the tRNA, initiator tRNA. And this tRNA would now initiate from UAG and would confer chloramphenicol resistance. Now, no surprises that when we touch the 3 GC base pair and change the sequence to the sequence that is found in the elongated species of methionine tRNA, the tRNA, of course, does not work here. So here is the system where we have our reporter cat gene, and this is the tRNA gene where the 3 GC sequence has been mutated, even though it is still called here 3 GC. Genetics nomenclature is somewhat strange. So when we say 3 GC, it really means here that this is mutant, and it does not grow on chloramphenicol. So basically, the tRNA is not functional in initiation, and we of course know that the tRNA is amino acylated and tRNA is also formulated. It binds to IF2. Everything is fine. It just does not initiate. So this is where I started my sort of work in India, and the work was again going back to very simple methods. In fact, this is another lesson that Tom gave me, and that was that, Umesh, you go to India, you do anything, if you see a growth on the plate or whatever, next morning, that should be a very good experiment. And this is really what I started out with, something very simple. And this was really facilitated by postdoctoral fellow Dinesh in the lab. And what I thought I will do is I'll talk to you about a little bit of these two strains. But these are really the suppressor strains. What this assay system that was set up was that we know that when we have mutation in the 3 GC base pair, it does not initiate from UAG, and the cells are actually chloramphenicol sensitive. But now if we make mutation in the E. coli genome, and if this mutation is able to rescue the defect of 3 GC base pair, it would start from UAG start codon and confer chloramphenicol resistance. All we had to do was rule out some of the unwanted mutations in the genome and then select for the ones which require this mutation, this tRNA, and this reporter. When the three things are required together, we have these strains. Now we've been working on these strains, for last many years really, and we have learned quite a lot about the process of protein synthesis. And what I can give you a little bit summary here is that if we look at why formulation is needed in case of initiator tRNA, there are two things really. One, it of course facilitates binding of the initiator tRNA to IF2. It increases the affinity of initiator tRNA greatly for IF2. The second thing, we've been able to sort of connect it is that there is actually a connection between the formulation and the 1-carbon metabolism. This has been told to us by our suppressor strains. So 1-carbon metabolism, if students would remember, it gives rise to 10-formyl tetrahydrofolate, which serves as a cofactor for formulation. And the 1-carbon metabolism also makes methionine. And therefore, both formyl group and methionine. So formyl methionine is really important. And you would want to initiate protein synthesis only if your cell is in a good energy state. So I think it's a really good checkpoint to start protein synthesis only when the cell is rich. The other thing that we've been learning about is the role of 3 GC base pairs. One of the things that 3 GC base pair does is as follows. When the initiator tRNA actually binds to 30S, it is not really binding in PP state. It is binding in a PI state And for it to go from pi state to pp state, it can actually fall off the ribosome. What we realized is that the 3 GC base pairs are important for this transition from this pi state to pp state. And we also realized, based on some of the additional work from the suppressors, of course, that the 3 GC base pairs are also important in ejecting the initiation factor 3 from the ribosome. And so once you have those 3 GC base pairs, you can make a 70S initiation complex where the tRNA is in correct orientation. You can then also eject the initiation factors from this 70S initiation complex to give rise to 70X elongation competent ribosome. So 3 GC are really very, very, very important. So much of the importance of formulation seems to be right in the beginning of protein synthesis or initiation, and the importance of 3 GC base pair seems to be towards the end of or in the later stages of initiation. So I mentioned to you about those D4 and D27. A graduate student, Suman Kapoor, was working on those. And this D4 and D27 turned out to be a mutation in the promoter region of the initiator tRNA genes, where in the locus which codes for 3 tRNA genes. And with these mutations in these suppressors, you can see the promoter activity goes down to really nothing. And if we look at the levels of initiator tRNA in these strains, we find, now this isn't a mistake, actually she overloaded these 2 lanes to show you that there is a little bit of fmat1 is still make. So this locus actually makes fmat1, and so this is the wild type strain. You have roughly 3 times the tRNA compared to this one, but in these strains, fmat1 is nearly gone. And we can sort of simulate these conditions by making a knockout of fmat1, where you see that the top band is missing, or we can also make a knockout of the second locus where the bottom band is missing. So she actually did all of this, and she also showed that if we have this strain where the 3 tRNA genes have been deleted, this strain grows all right at 37, but when we subject it to 22 degrees, it doesn't grow that well. The wild type strain grows, but the mutant does not grow. Now she finished her PhD at this stage, but luckily another graduate student, Sunil, a very bright student, joined, and he found this observation quite interesting. He found it very interesting because, often the cold sensitivity is associated with a defect in ribosome biogenesis. And so ribosome biogenesis really occurs, it starts very early on. So we have a precursor transcript which has all of these, regions in the transcript. And as the transcription goes on, these transcripts are cleaved off. You have extensions to 5' and 3' of the 16S, which gives rise to your 17S rRNA, and that is then finally processed into the 16S rRNA. There are many ribonucleases that are involved, which are sort of listed, and I'll keep coming back to these. So you have then finally the mature 30S ribosome. So if we were going to probe for whether there is a defect in ribosome biogenesis, what he wanted to do was look whether there is accumulation of 17S rRNA. Now 17 and 16S migrate very close together on agarose gel. So what he did was he designed oligonucleotide probes which would anneal in the immature region of 16S, that is to 17S or to 16S or to downstream of the 16S. And he can then do the northern blot analysis, and one thing that you can very clearly see is that when you go down to 22 degrees, you accumulate a lot more of the precursor. And he can then take the ratios and come up with this kind of a plot where the wild type strain has that much precursor. As you delete the tRNA genes, the precursor levels go up. At 22 degrees, this really goes up. So he set it up as an assay system for him, and then he started asking questions as to what happens. Now, another observation that he made was that if in the same kind, if he takes the wild type strain of E. coli, and if he actually overproduces the tRNA, which has mutation in the 3 GC base pair, that also gives him the cold sensitive phenotype. And he can rescue this cold sensitive phenotype by overproducing a wild type copy of initiator tRNA. And so this also sort of went back at that there is again a biogenesis defect here. So whether we take away the wild type copies of initial tRNA, or we overproduce a tRNA which is defective in the 3GC base pair, he gets the same phenotype of cold sensitivity, he gets the same phenotype of ribosome biogenesis defect. Now in genetics you would also then want to combine the two and look at whether there is a synthetic effect, and this is precisely what he found. So when he takes the strain which is deleted for 3 tRNA genes and he overproduces the mutant tRNA, he doesn't find any growth at this one. So they were working together, and in fact he can measure that also. So that clearly said that the two are working, doing the same sort of things. Now one would have wanted to work with the situation when we delete the 3 tRNA genes and overproduce 3GC base pair, but that's a very, very tough situation. The bacteria doesn't like it, keeps coming back with suppressors, and therefore we didn't pursue that one. But then what he did, that he started looking at how this tRNA is participating in ribosome biogenesis. And one of the things very quickly he found that if he had the wild type anticodon in the tRNA which has mutation in 3GC base pair, it would cause biogenesis defect. So here we have the sector 3 and 4, you can see this one, that there is growth is weaker. Now if he changes the anticodon from wild type to CUA such that it will not normally participate in initiation, that defect is not seen. So here is a situation where he has changed the anticodon, and sector number 5, the growth is quite well. So he figured out that there is actually involvement of this tRNA in initiation, and that must be connected with ribosome biogenesis. Now the question was, is it really so? And then we went back to some of the earlier work that another graduate student, Smriti Arora, had done in the lab. And what she had done was, soon after when he published this paper of crystal structure, how much time do I have?
You have about 10 minutes.
Oh, great. So what she found was that ribosomal protein S9, there are, this C-terminal tail of S9 is actually going and contacting the 3GC base pair. So we have the P site here, and this region right here is 3GC base pairs. This tail is contacting it. Also, there are a couple of methylations at 966 position and 967. These methylations also sort of make the contact with initial tRNA a little bit tighter. So if we lose these methylations and lose the C-terminal tail of the S9 protein, by the way, this S9 tail 3 amino acids SKR are highly conserved. So when she deletes this, she finds that again there is compromise in initial tRNA binding, and in fact there is cold sensitivity. We didn't connect it at that time, but Sunil looked at it, and he also then decided that he will try to combine his 3GC mutation with these mutations, and also with another thing which is RBFA, which is a bona fide ribosome biogenesis factor, with the 3GC, and this is what then he found. So he takes this S9 delta 3, and you can see that there is actually further defect in the growth. But he finds with RSMB and D, these are the methyl transferases which methylate 966 and 967 and 966, and you see that there is a compromise in the growth. But when he combines with the RBFA, there is no defect. And you can see that RBFA, you accumulate a lot of precursor. With RSMB and D, you start accumulating more. But with introduction of the 3GC base pair, there's really no change. So what this really meant was that the 3GC base pairs must be working downstream of RBFA. So when you have RBFA defect, 3GC is not coming in the picture. So 3GC role has to be downstream of RBFA. RBFA is working very early on. So 3GC must be working downstream. So then the other thing that he did was he then started looking at the combination with various RNases that are involved in processing of this 17S rRNA. And so many of these strains are available. So he just combined this 3GC plasmid he took into these strains, so that if there is actually a load, increased load on RNases, he would now see a processing defect, and he did see. So he sees in RNase B, R, and G, 3 of them. And that is shown in the growth also, that in the control, the strain is going like this, it goes flat. So this is one way of looking at that there are actually role connection between the 3GC base pair and RNase, and he can do a complementary experiment, and that is he can now overproduce RNases into this system where the 3GC plasmid is already there. And you can see here again that when he brings it at 22 degrees, here is the sort of control where he has the plasmid, 3GC plasmid and the vector. One doesn't see much growth. But when he brings in the RNase P, RNase PH, the growth starts to happen. So all of these RNases that we were talking about, RNase R, one can start seeing the growth. So again, there is a connection between 3GC and RNase R. Now the point is, the initiator tRNA is doing two things. We are saying that it's involved in biogenesis, but one can argue that we are affecting initiation of protein synthesis also, which means we might be depriving the cell of some ribosome biogenesis factor. Now, which one of these is correct? And what Sunil then did was he decided to do an experiment, old-fashioned experiment, and that is he took the cells, put rifampicin. Now, rifampicin is going to block any new transcription, and if he can see that the processing continues, that means in most likely situation that the new protein synthesis is not required. And this is precisely what he saw, that if you have here the control, so let's talk about these 15 minutes and 30-minute control, which are not treated. Now you treat with rifampicin and incubate them for 15 minutes and 30 minutes, the precursor levels keeps coming down. We can also incubate, while we are incubating the cells, the antibiotics that inhibit protein synthesis. This processing is not interfered with casipromycin, spectinomycin, streptomycin, chloramphenicol, fusidic acid, puromycin. None of them affects the processing. The only thing that affects is tetracycline. Now, tetracycline also is known to bind to ribosomal P site. So it seems to be competing with the initiator tRNA. So this then suggests that the initiator tRNA is doing a job which is more in the ribosome biogenesis than in the protein synthesis. Had that been the protein synthesis, we would not have seen this processing happening, especially in the presence of rifampicin and the antibiotics that inhibit protein synthesis. Then the question, of course, was that whether it is at the 30S level or at the 70S level. Now this again, he did the experiment where he fractionated the ribosomes or the polysomes on sucrose density. We can isolate the peaks 30S, 50S, 70S and polysome, and now find out again which of these population accumulates more of the precursor 16S. And he found that in case of this particular strain where we are overproducing 3GC tRNA or the mutant tRNA, we accumulate more into the 70S. So here is the vector. You hardly accumulate any precursor in the 70S. With tRNA, wild type tRNA overproduced, we don't accumulate much. But in this particular strain where the mutant initiator tRNA is being produced, we accumulate precursor 16S rRNA. So it is, and we've done a few other genetic experiments that I'll spare you with the details of those. But all of that then led us to propose a model which follows us like. So we have the precursor 16S rRNA in 30S. This is capable of binding initiator tRNA. It can be joined by 50S subunit. At this point, initiator tRNA does something which then allows the ribonucleases to clip off the final terminals of the 17S rRNA to give rise to the mature 16S rRNA and then go on to protein synthesis. So this would probably be happening only during the pioneering round of ribosome biogenesis. Now the point that can be asked is, this is our completely artificial system. Would it have any relevance whatsoever to the in vivo situation? Now the only way it can have any relevance would be if there are situations where the levels of initiator tRNA can be altered. So this there are situations that are known which cyclic AMP cap, in fact, it binds to a downstream gene, RG, but the promoter of RG is sort of overlapping, or the cyclic AMP binding site is overlapping with the promoter of MAT-Y. And this binding actually downregulates. Again, you can see energy connection here. There is also a connection that the promoter of the gene, where the locus, which has 3 tRNA genes, is downregulated by PPGPP. There is a very interesting system that some of the viruses, in fact, come with endotoxins, WebC here, which is a ribonuclease, and this guy targets just the initiator tRNA, looks for 3GC base pair, targets that one. So there are situations in the cell which can downregulate the level of initiator tRNA. If that, then could the heterogeneity at the 3' end of the 16S RNA be of any use to the cell? That is the question that arises. Now, the answer to this, we sort of started looking. There's a paper published by Harry Noller, and what he has shown was that you have in the 16S rRNA helix 28, helix 23. The anti-Shine-Dalgarno sequence is somewhere right there. When you make a pairing with the messenger RNA, these 2 helices actually sort of go away. And depending on the extent of the base pairing and the placing of the base pairing, one would imagine that this displacement would be different. Now, why am I talking about this displacement? There are a couple of residues here, 1338 and 1339, which are right next to this helix 28. And these 2 residues, 1338 and 1339, interact with the 3GC base pair. So there is going to be a direct connection there. So then a student in the lab began to change the extent of the Shine-Dalgarno and anti-Shine-Dalgarno sequence interaction. And what I can actually show you from this slide right here, the moment he goes into base pairing in the immature region, this whatever shown here in red is actually not present in the mature 16S. You can see that there is increase in protein synthesis with the mutant tRNA. And again, you can compare this versus this. This is completely mature. This is immature. There's increase. So it's quite possible, and of course we are looking for this now, that under stressful conditions, which let's say downregulate initiator tRNA in some way, there is heterogeneity that is created in the ribosome, and that heterogeneity is useful for the cell to, in fact, come out of those stressful conditions, and sort of, there is a change in the proteome, and that can be done. So, of course, a few other pictures that I found. Now, the acknowledgements really, this work was begun by Suman Kapoor, Smriti Ahuja. Sunil Shetty really took it to a conclusion, and Riaz is continuing with this. And the laboratory is supported by funding from Government of India sources. And thank you very much for your patience.
We have time for a couple of questions. Yeah, Richard there.
I just want to know what is that I site?
I'm sorry?
I position.
I?
On the ribosome.
Oh, I position is towards the E site.
But is it a transitional site? What is this?
So it's a sort of a, during translation, of course, you're familiar with the hybrid states, which are, let's say, AP site or PE site. In this particular case, this is really not really a PE site, but it's towards the E site. So it's called as PI site. This is the term coined by Jody Pugliese. So it's in PI site. It's not really oriented in a place where the acceptor stem will find a position in the P site of the 50th.
So how does the tRNA then move to the real P site?
So that's where I believe the way it is happening is that the 3GC base pairs are interacting with G1338 and A1339, and that interaction is really what is involved in the conformational change, which allows this tRNA to go into the PP state. Well, both are connected anyway. Yeah.
I have a question. So
Yeah.
this was done in bacteria, correct?
Yes, yes.
So does this apply to mammalian cells?
Good question. The only thing I can tell you about that is that, people have measured the levels of elongated tRNA and initiated tRNA under cancerous state. And I believe they find that, the relative levels of initiated tRNA go down there. So I think this may
Actually, we
find some
found that tRNA is double in cancer cells in general.
Initiated is more?
Yeah.
Okay, so there are some reports which I think, but, that again will be relevant from,
Right.
change in the, yeah.
Right. Anyway, any more questions? Yeah.
Sir, very nice work and very good presentation. Sir, you have done a lot of work on ribosome biogenesis.
Some work, yeah.
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 So my question is a kind of layman question. There are two kind of ribosome, as we know, that eukaryotic and prokaryotic, and they are quite different. Even the ratio of RNA and protein are different. The prokaryotes have 60% RNA and 40% protein. Eukaryotic have 40% RNA and 60% protein.
I think you're talking about mitochondrial.
Yeah,
ribosomes.
this is
Yeah,
what I'm
yeah.
saying. So means within the same cell, we have these two kind of ribosomes, the prokaryotic and eukaryotic in cytoplasm and mitochondria. What is the need of having these such a complicated ribosomal systems in a cell?
That I will find difficult. I think Tom can only answer that. Tom. I think you require some of the proteins to be synthesized within mitochondria, and perhaps I think it's difficult to take them from cytosol to mitochondria. And I can only speculate. So I think you need to make those proteins away from cytosol into mitochondria.
Thank you.
Okay, any other questions? All right, with that,
Thank you.
thank you so much. Oh, I'm sorry, one more.
It's not a question to Professor Varshney, but it's only a addition to the question asked previously. It's very clear the evolution of the organelles, mitochondria and chloroplast, and that tells you the answer.
Yeah.
Okay, with that, thank you very much, Umesh. All right, our next speaker is Matthew Sachs. He's in the Department of Biology, Texas A&M University in the US. And he's going to continue this theme. So he's going to talk about gene regulation through the control of ribosome movement.
All right. Well, thank you, everybody, for attending, and thank you to the organizers for putting together such a beautiful conference. I'm going to tell you today about translation initiation in eukaryotes, and I will focus experimentally, of course, largely on work that has occurred in my laboratory and the laboratories of my collaborators over time. I think you can see here first the main subject of the research, which is the fungus Neurospora crassa, and you can see how rapidly it grows. This is time in hours. It grows millimeters per hour. And I show this slide because, in fact, can the lights be reduced is the question. Yeah, it is pretty bright in here, I agree, looking at... Okay, not a question. Is that better to see? Yes. So this is classic growth of a filamentous fungus. This is an ascomycete, millimeters per hour. And you can see it's now sporulating. And in fact, when I was a first-year graduate student, Marcia talked about her decision to choose a lab. This is an image, almost another kind of a time lapse, right? This is probably the early 1980s, and just a close-up, of course, there's Gobin and Tom, and I joined Tom's lab